Howdy, welcome back. We are moving on in our journey through the history of microbiology. So we've covered spontaneous generation, we've covered fermentation. So now we're going to turn our attention to disease. So this next video is going to be talking about when people figured out what actually causes disease, and specifically infectious diseases. So this doesn't account for like cancers or inherited diseases, but more of infectious diseases, which were early 1900s was the #1 leading cause of death for all age groups, especially children. So it was more likely for you to die of an infectious disease like influenza or cholera or chicken pox or measles, any of those a disease that's caused by a pathogen like a bacteria or a virus or a fungus. You're more likely to die from that than you were old age. Or heart disease, or cancer, or diabetes, or any of the things that we now see are the leading killers in our society. Infectious diseases aren't as prevalent as things to kill you as they used to be basically in the invention of antibiotics and better medicine. And we're going to talk about all the things that, how we prevent disease in the next video. So I'm not going to give anything away. So prior to this next guy we're going to talk about, Koch, is just like before spontaneous generation was put to rest, the idea of what caused disease was a wide array of things. It could be your parents sinned, and that sin was passed on to you, and so that's why you're sick. It could be that you lived close to a swamp and you were exposed to bad air. If you've ever heard of the disease malaria, air, that's what malaria, bad air. They did not know it was spread by mosquitoes that lived near swamps. They thought it was the bad air, the mucky air that kind of came off of the swamp and the wetlands that gave you malaria. Isn't that interesting? Like, yeah, malaria, it's a bad disease caused by mosquitoes, but no, it's named after what they thought it was bad air. So there was no connection between a pathogen like a bacteria or a virus and a disease, because remember, pre-Leeuwenhoek, bacteria didn't even exist in the known world. So how could they attribute disease to something that they didn't even know existed? But once bacteria, viruses not until the 1900s, but until we recognized that there were these invisible critters living in our world, there was no reason to say that they were the cause of things. But once we identified them, then we could start figuring out, hey, these are small enough that maybe they could do something to us and cause disease. So just some vocabulary here going through the notes. We have a pathogen. I've mentioned that word a couple times already. A pathogen is just anything that can cause disease. So for the most part, it's going to be bacteria or viruses, or some fungi can cause disease. Protozoa, like malaria, is caused by a protozoa, a eukaryotic protist. And there's not a lot of animals or plants that cause disease. So it's mainly those microscopic things. A pathogen is just anything that causes disease, usually a microorganism. And then the next word is called etiology. So etiology is the study of the cause of disease. So you study how diseases are caused. So that's etiology. The main guy here in our story is Koch. And so he's, again, late 1800s, about the same time Pasteur was doing all of his work. I want to say Robert Koch? That doesn't sound right. He's German. So K-O-C-H, not Coca-Cola Coke. All right, let's take a look. Oh, Robert, yeah, I know. So he has a whole chapter in the Micropunctures. Really, really great story if you are interested in this, scientific history of science. Excellent book. Once you get done with school, read it for leisure. It's very awesome. So the story goes, Robert Koch, he was a country doctor. He went to med school, but he really just liked to be out in the country, going from house to house. He helped people, he helped farm animals. He had his own microscope. You can see that there in the drawing, the picture. And he just had a really good curiosity about things. And again, as all discoveries usually do, he started with some observations. So livestock were susceptible to a disease called anthrax. And it was really devastating for farmers and ranchers because they could lose a lot of their livestock to anthrax. So he did some experimenting. And I might be missing some parts of the story, but if you want the whole thing, you can read it in the book. So he took some blood or sputum or, some bodily fluid from an animal, probably a sheep or a cow or something that died of anthrax. And he got some rats or mice, some kind of lab animal, even though it was probably just his kitchen or his barn. And he took a splinter of wood and he stuck it in the blood from the infected animal and he poked it into the tail of the rodent. I know there wasn't any animal rights back then. And what he noticed was every time that he exposed one of those lab animals to this infected blood, they got sick. And not only that, but he dissected these animals and he looked at their tissues and their blood under the microscope. And every single time he looked at an infected animal's blood or tissues under the microscope, he would see this thing, these tiny little stringy-like things. And he didn't see them in any healthy specimens. So he would probably be collecting specimens from farms or, his rat collection or whatever. And he never saw that particular thing in the specimens of healthy animals, but he would always see it in the specimens of the unhealthy animals. So he made this conclusion that wherever you have this particular organism, that's what causes the disease, not bad air, not, walking backwards at midnight or any of these weird things. It was the presence of this organism is what caused disease. So he came up with what we now call Koch's postulates. He could have called them that. I don't remember if he named them after himself or if it kind of came later. So there's four, kind of five-ish, 4 or 5 key things here. I have 4 in the notes. So the first one is your suspected pathogen, a thing that you think causes disease, has to be found in all cases of the sick organism and absent from all cases of the healthy. Because if you find that pathogen in healthy organisms, then that's not a very good candidate for being the thing that makes things sick with that particular disease. Okay, so that was our first one. It has to be found in all sick organisms and absent from healthy organisms. Then you must isolate your suspected pathogen from your sick host. So in this case, it's a dead mouse, but not all illnesses make you die, so you'd be sick. So you are going to isolate them, and so you're going to grow them on a petri dish and identify them under the microscope. So that's the one I'm interested in. And then the third postulate is that you are going to find a healthy susceptible host, and that's important because susceptibility means a disease that can make a rat sick, you have to use the rat as a susceptible host. If you use a human or a dog or a chimpanzee, they may not be susceptible to this disease that affects rats. So to test the postulate, you need to keep it in the same host, susceptible host. So healthy susceptible host gets injected with your suspected pathogen. Then step 4 is that healthy susceptible host gets sick with the same disease, right? So same signs, same symptoms, and isolate. So I'm kind of making these be 1. This is my number 4. So the healthy susceptible host succumb to the disease and you re-isolate and identify the same organism. If you can satisfy all 4 of Koch's postulates, then it is a good indicator that that particular pathogen is the cause of that particular disease. Okay, so that was Robert Koch's contribution to disease, is making that connection between microscopic organisms and their ability to cause disease. The next individual on the last slide here is taking a look at Christian Graham, which again, we're in the late 1800s, so all about the same time frame. Not a lot of time has gone by from Pasteur to Koch to Christian Graham. So Christian Graham, his contribution, which we use in microbiology, it's like the #1 staple of staining techniques, and you'll be doing this in our lab kits, is he developed a differential stain, which means you use multiple steps, different dyes to stain the bacteria. And what he came up with is if you follow these particular steps of what we now call the Graham stain, named after him, is that certain groups of bacteria stain purple. We call those gram-positive bacteria. And after the same 4 steps of doing the Graham stain, another different kind of bacteria stain pink. And so these are called gram-negative bacteria. And that was, he didn't know why yet. We're going to be learning about that in chapter 4. But it's a structural difference between the cell walls of gram-positive and gram-negative bacteria. But what this allowed him to do, it gave us better identification purposes for those potential pathogens. So where Koch was looking at them under the microscope, Christian Graham was able to take that specimen from the sample, maybe grow it out, stain it, and now we have not only the appearance of what it looks like in the microscope, a little stick or a ball or a spiral, but now we can add to that it's a gram-positive organism or it's a gram-negative organism, because there's, this is a bacillus, which is a rod-shaped bacteria. These are bacillus. They're also a rod-shaped bacteria. So if you didn't have staining and everything looked the same under the microscope, it would be really hard to tell, is that really that organism that I think causes the disease? But then once you can add some differentiation to that, I don't have pink in my palette here. So once you can differentiate those, then you can be more confident in your differentiation between this is the bacteria that causes these because it looks like it, it's found in all the specimens, and it's gram-positive. Because if you get a gram-negative showing up, it's probably not going to be that one if all of the other samples shown gram-positive. Okay, so that was Christian Graham's development of the Gram stain. All right, so off to the next lesson on how do we prevent disease now that we know what causes disease, infectious disease. Okay, I'll see you next time.